Hi everyone, I'm Grant, and I'd like to tell you about a few roles bioinformatics plays in target identification and validation, in biomarker discovery, clinical trial design, and precision medicine. This isn't intended to be comprehensive, and there are entire areas of the drug discovery and development process we won't touch. In particular, structural biology and modeling can play a role in target identification, and even more so later in the drug development process. Our focus at the Bioinformatics CRO is omics, hence this talk will be very omics centric. And please excuse my voice, I have a bit of laryngitis. So it's crucial to start with the right drug target. 57% of phase 3 clinical trials that fail do so for lack of efficacy. There are a variety of reasons for this that can sometimes be difficult to pick apart with confidence, but in a well-designed and well-powered study with robust target engagement, this can sometimes reasonably be ascribed to having picked the wrong target, especially when many such programs around that target fail. There are estimates that it costs over $2.5 billion on average to take a drug to market, considering all the failures that have to occur for a success. Let's talk about macro factors in the biotech industry for a moment. Biotech is a bit of a jackpot industry. Most programs fail, some of these after substantial investment. Similarly, most biotech startups fail. So for the industry to be sustainable, companies that are clinically successful need to be sufficiently profitable to offset the losses elsewhere in their investors' portfolios. For the industry as a whole, returns in biotech are comparable to some other sectors that have considerably less risk. Obviously, there is substantial political pressure in the US on drug pricing, and that puts a ceiling on the revenue side of things. Meaning, all this points to the need in the industry for de-risking programs early, one element of which is improved target selection. So I don't mean, I don't want it to come across that, bioinformatics is some kind of panacea for biotech or for target identification. But there are elements of the target identification process that could be improved. Informed target selection involves many disciplines spanning science, intellectual property law, and commercial considerations. Competitive landscape matters. Entering a crowded space, competing against companies with a head start and far greater resources may not be the best move. Freedom to operate is crucial in biotech. If others have patents covering how you would plan to drug the target of interest, and it's not an option to license these, you may not have a great target. The target needs to be druggable. It needs to be safe. There needs to be a potential for meaningful improvement over the standard of care and at a reasonable price point. We could go on and on. In my opinion, failing to involve bioinformaticians in a target selection process is negligent. I've listed a few non-exhaustive examples here. Bioinformaticians would need to be involved in the design and analysis of functional genomic assays. For example, bit.bio discovery, who's a partner of ours under my other hat at bit.bio, runs CRISPR screens on in vitro models of disease. Model systems are great, but there are always questions of relevance, which is where human genetics shines. Programs around targets with human genetic evidence are twice as likely to show efficacy as those that lack such evidence. Bioinformaticians can also help you understand where, when, and in what pathophysiological conditions your target is expressed. Finally, the best approaches use both public and proprietary data to flesh out the most complete landscape of the target. Platform companies that have the ability to drug a broad class of potential targets need to choose among them, and additionally need to narrow to the indications of interest. This target indication matching is also a multidisciplinary effort, particularly for selecting the indication. For bioinformaticians to bring value to this process, the broad search space means the efforts need to scale. which typically involves collating data across a large number of candidate targets across features that relate to a number of indications. Resources such as Open Targets, UK Biobank, Japan Biobank, TCGA, and various gene expression databases can be sources for important phenotype information that can help progressively narrow the search space as more refined hypotheses emerge. With a narrower set of questions, one can then dive into better powered but more specific genetic datasets, case control functional genomic datasets, and so on. The plot here shows some results on genotype-phenotype associations from exome sequencing in UK Biobank recently published, providing an unbiased look into traits associated with genetic variants in humans. It's important to understand the expression of your target of interest. How does it vary developmentally and through maturation? What happens in aging? Does it vary across cell states? Is it only seen in certain pathological conditions? Does it potentially modulate key cell-cell interactions? For example, as a receptor of a ligand that primarily is found in pathological cell states. For this, one can draw on bulk reference data sets, such as Phantom, GTEx, and Human Protein Atlas. Single-cell RNA-seq data sets can be quite informative here. There's a growing set of reasonably comprehensive examples. Human Cell Atlas, the Fetal Cell Atlas, also called Descartes, Human Cell Landscape, Tabula Sapiens, and several in mouse, if you're thinking ahead to your preclinical in vivo work. We've not even talked about epigenetic data, but depending on the nature of your target or mechanism of action, that could also be critical. Models are models. They are not the disease. Bioinformatics can help better understand the nature of your model and what aspects of the disease they reflect, or importantly, do not reflect. Transcriptomics, comprehensive studies of gene expression, is one reasonably cost-effective way to start to get some insight into molecular processes that are perturbed in case control studies of patient tissues as compared to your model. WGCNA, weighted gene co-expression network analysis, is a powerful way to look at this. With the right sample sizes and replicate structure spanning the necessary conditions, you can identify modules of mutually co-expressed genes and then annotate these post hoc on the basis of their expression patterns, constituent genes, and functional enrichments. Once you have some idea what known biology these co-expression modules reflect, you can then look at the preservation of these modules between disease and the disease model, and at the similarity of their disease-associated patterns between the two. Does it go up? Does it go down? So on. You've then defined a molecular phenotype that can be rescued like any other phenotype, and you have greater confidence that this is the same phenotype seen in the disease, which is especially good if this is in a genetic model with the same mutation as patients. So then you have a robust conserved phenotype downstream of a shared molecular cause. Target validation is often done in the context of a model system, but the translational relevance will be much greater if the target is effectively validated in patients. Genes containing variants that modulate the risk of developing a disease may accumulate chronically over a long period. Most genome-wide association studies are designed to look at this. These can be linked to molecular QTLs and such to understand directionality and to start filling in some understanding of the intermediate physiology. However, the translational relevance of these is perhaps a bit less than variants that impact disease progression, prognosis, outcome contingent on diagnosis, and so on. This is most analogous to what one does with a drug, which of course is typically given after one is diagnosed with a disease. Bioinformatics also plays a key role in precision medicine. How does one differentiate responders from non-responders to a specific drug? This can inform what is prescribed to whom. If one has an in vitro assay with, functional genomic readouts, what features are most predictive? There's often a trade-off here between the interpretability of a model, which helps with biological understanding, and the performance of a model, which may be more important for real-world application, so long as it's robust and not due to overfitting. Proprietary data is often important for this, but resources built on the cancer cell line encyclopedia, such as the Cancer Dependency Map, or DEPMAP, are example public data sets that are useful for this. For example, CCLE data can be used to identify shared properties of responsive or non-responsive cell lines. DEPMAP uses CCLE to call out shared characteristics of cell lines that are sensitive to knockouts of various genes. As usual, the cancer field leads the way in functional genomic resources, but there's no reason one can't apply these approaches to other classes of indication. First, you need to understand the genetic variation in your target of interest. What interesting variants are present? How do they co-associate and segregate into haplotypes? We use 1,000 genomes for this. Once haplotypes are defined, what are their frequencies across populations? This can be important for clinical trial recruitment if you decide to target a particular haplotype or group of haplotypes or carriers of those haplotypes. It's important to layer on known functional annotations. For coding variants, how do they impact the amino acid sequence, and how is that predicted to affect the protein? Are the variants expected to impact splicing? What disease or phenotype associations are there? Is there co-localization of these phenotype associations with molecular QTLs? For example, gene expression, protein levels, transcript isoforms, epigenetic modifications, and so on. If there are variants that impact, say, the activity of an enzyme or other biochemical properties, these need to be layered on as well. Then you can use these human genetic data to inform clinical trial design. In oncology, TCGA gives a sense of what's out there and can be used for genetic-based survival stratification, looking at germline and somatic variants, which could potentially be used to identify candidate groups of patients who may benefit the most. There's also the potential to narrow to a more mechanistically homogeneous group of patients by stratifying, for example, on a major genetic risk factor or a particular multi-omic profile. We've already discussed WGCNA, so I won't linger here for long, but I'd like to note that WGCNA can be applied to generate hypotheses around mechanism of action where that is unknown. It can be applied to bulk or single-cell RNA-seq, and the molecular effects are typically more interpretable than traditional differential expression, where the differences are all shoved together. Bioinformaticians can also play a role in discovering biomarkers for target engagement. For example, an omics readout from peripheral tissue in drug versus vehicle-treated animal models can help identify candidate readouts that are plausibly connected to the mechanism of action. One can then test if this peripheral readout varies coherently in human data sets, and given patient data, one can also see if it varies with disease. In the ideal case, one derives a robust and coherent panel of markers shared across species. These are just a few applications of bioinformatics in the drug discovery process and beyond. Many small biotechs don't have bioinformaticians, but they should, and I hope they picked up some at the job fair today. I'm always a believer in returning time, so let's wrap it up and open the floor to questions.
Yeah, thank you so much, Grant, for your excellent talk. Yeah, so one of the common themes of this conference, I think, has been differentiating between like biologists using bioinformatics and bioinformaticians conducting bioinformatics. And so we've talked about a lot of different tools that are designed for either bioinformaticians or for biologists. So you work a lot with biomedical researchers and obviously also bioinformaticians, but what are some common misconceptions that biomedical researchers have about the work that you do and the benefits that bioinformatics can have on their research and drug discovery?
So I imagine most bioinformaticians have the experience of the misconception that, bioinformatics is pressing a button, right? Why can't you press your button and get this to me today, kind of thing, right? Another common thing we see is non-bioinformaticians not understanding the importance of standardization in the input, right? We often get input files that are manually created, and each batch is named in a different way, requires different cleanup, and so on and so forth, which obviously for any kind of automated processing is a real mess and can in some cases take a considerable amount of time to figure out what's going on. I think there's also often misconceptions around how things scale. A number of times we've had people think it was easier for us to provide a subset of results than the full results. And to think that providing that would scale linearly, that if you provide a fifth of the results, it would be 5 times as fast as providing all the results, right? Because there's I think people often have a mental model of running an assay and you're pipetting 1 plate versus 5 plates versus 20 plates or whatever, and obviously that's not the case in what we do. We find there's also often kind of misconceptions around projects that are development heavy, and kind of not a good understanding of the software development process, for example. Also, sometimes bench scientists will ask for safe features, and one feature may be trivially easy to add, and one feature may require, considerable re-engineering. And I would say there's often not a good understanding that 2 things that can look from the outside to be quite similar, kind of under the hood can be so different. So one thing we focus on at the CRO is client education, and whenever we work with bioinformaticians and we're basically handling their overflow, it's not necessary. But sometimes when we work with bench scientists, a bit of that is to kind of let them know, like, what's the most cost-effective and efficient way to work with this. One thing we see sometimes from academics, we tend not to see this from biotech clients, is sometimes not much thought put into what's work for professional bioinformaticians and what work is better for my graduate student to do, right? If you're adding colors to an Excel spreadsheet or something, have your graduate student do that. It doesn't make sense to have a bioinformatician do that. And so, we sometimes push back on that. I think another common thing that bioinformaticians run into is, especially when working with academics, is this concept of payment in the form of co-authorship. But co-authorship doesn't pay the bills, right? It's honestly kind of insulting, but it's a very common offer I think people receive. Yeah, so I think those are all quite common. I'm sure there are a lot of others that aren't springing to mind immediately.
Yeah, certainly. Yeah, and so with this growing accessibility to bioinformatics and people getting access to open source tools, what do you think are some indications that more of a bioinformatician's touch, if you will, is required? Like, what are some indications that people may need to seek further assistance in a bioinformatics project?
Right. So I'm a big believer in providing, easy to use tools to reduce the need for kind of computational biologists to come in and help with projects. But I do think those tools sometimes need guardrails, right? Very often, these tools are used in inappropriate ways to do analyses that frankly don't make any sense. And some of that you can mitigate through documentation and warnings and things like this. But I think tools are great for sorry, I looked over at the chat, got distracted. Yeah, I mean, tools are great for empowering bench scientists, but they do need to be used appropriately.
Definitely. Well, Grant, yeah, thank you so much for joining us. It's been a pleasure to host you, and yeah, I'm gonna turn the mic over to Adam. So yeah.
Fantastic. Great.
Yeah.
Thanks.